Good morning, everyone. Good morning, everyone. It is really my pleasure to introduce today's speaker, Professor Meng Guangxun, an expert in the fields of mucosal immunology and inflammation. Professor Meng is the chief of the unit of mucosal inflammation and the executive director of the Center for Microbes Development and Health at the Shanghai Institute of Immunology and Infection, formerly known as the Institute Pasteur of Shanghai, Chinese Academy of Sciences. Professor Meng earned his PhD from the Technical University of Munich in Germany in 2004. He then moved to the United States, where he joined the NIAID at the NIH, as he just mentioned briefly, as a postdoc fellow in Dr. Warren Strober's lab. After completing his postdoctoral training in 2010, he started his own independent research group in Shanghai. His team focuses on understanding mucosal immunology and inflammasome activation in barrier organs, including the gut, lung, and the skin. Specifically, his research explores the immune responses to viral infections in the respiratory system, as well as the role of inflammation and the immune system in cancer within the gut. His groundbreaking work has been published in prominent journals, including Nature Immunology, Nature Communications, PNS, etc. Um, we are thrilled to have him here today to share and hear his latest insights about his recent findings. Please join us in welcome Professor Meng Guangxun. Please.
Okay, thank you, Paul, for the kind introduction. And I also would like to take this opportunity to show my appreciation and thanks to Chuan for organizing this series of great seminars since these years. I attended quite some of them and learned a lot. So it's really a very nice platform for immunologists, especially yeah, um, the Chinese immunologists share knowledge and communicate with each other. So yeah, today I want to show some of my recent studies as well as some published work. Uh, yeah, hopefully get feedback from colleagues and yeah, possibly we can initiate some um, further communication and collaboration. So I'd like to start with this general slide to show that in the world we are living in, there are a lot of different microbes, including many types of bacteria, viruses, and also fungi. Our immune system was taken as the front line or the key settings for the host to combat against pathologic microbes. Um, but in the numerous types of microbes, the really pathogenic ones are very limited. Usually, a lot of microbes form a beneficial relationship with us, that's known as commensal microbiota, which is studied during the recent yeah 10 or 20 years. So we offer a good niche for the commensal microbes, and they can digest special substances and provide essential nutrients for us. Um, on the other hand, in the mucosal organs, like in the gut and the lung, so there is a constant interaction between commensal microbes and our immune system, which is very important to keep the homeostasis locally. And also, there is communication between the mucosal organs and the mucosal organs with other remote organs through the molecules generated either from microbes or from the immune system in the mucosal tissues. So this is very important to keep the homeostasis in different organs. So no matter it's pathogenic microbes or commensal microbes, the interaction between microbes and the host needs a molecular intersurface. Uh, yeah, in most cases, products from the microbes, now called microbes-associated molecular patterns, can be sensed by pattern recognition receptors expressed on host innate immune cells or epithelial cells. This recognition initiates signaling and leads to either quick innate immune response or instruct of adaptive immune system to establish um, immune memory. So pattern recognition receptors have been studied quite extensively during the past 20, 30 years. Now, many different members have been identified, including toll-like receptors and also NOD-like receptors. Here, a group of NOD-like receptors and some of the DNA sensing receptors can form a special protein complex called inflammasome. And assembly of the inflammasome will lead to activation of caspase-1, and caspase-1 can cleave pro-alpha beta into its mature form and fulfill the downstream function. So far, quite some inflammasomes have been identified. One function of the inflammasome activation is maturation of beta and IL-18, as I just mentioned. Also, it leads to the gasdermin cleavage and proptosis. Most inflammasome members have specific ligands like AR2, M2 recognize DNA. But there is a special member which is NOB3. NOB3 inflammasome can be activated by many different substances. And also, there's another special property of the NOB3, which is there are a lot of gain of function mutations identified from the NOB3 NOD domain, which cause to a set of autoinflammatory diseases called quarter-associated periodic syndromes. When there's a gain of function mutation, the NOB3 inflammasome activation doesn't need two signals, but can be activated by one pump stimulation like LPS. Back to quite some years ago when I was still working at NIH, we generated NOB3 mutant mouse mimicking the CAPS patients. So macrophage cells from this animal do not need the two-signal system to activate, but only one LPS stimulation can lead to activation and alpha beta generation. So these animals do not only have higher alpha beta expression, but alpha beta also drives the Th17 development. So this was also one of the few studies showed that alpha beta is an important element to drive Th17 cell development. Following this work, we found that if the pregnated mutant mouse is treated by antibiotics, the pups have much less skin inflammation, which means the skin inflammation in these mutant animals is associated with microbes. So we immediately realized that in our body there is a lot more microbes in the gut, but I observed at that time the gut of this mutant animal is totally normal. So I followed this question and did a set of studies. Quite a few years ago. That's to understand how come this phenomenon happened. So we used a reporter mouse showing the expression of NAP3. First, we want to know whether there is NAP3 expression in the gut tissues. And we found that in the gut epithelial cells, there is no NAP3 expression, but in the innate immune cells in the gut, including the macrophage, different types of dendritic cells or monocytes, there is a clear NAP3 expression. So it is expressed in the gut innate immune cells. But why there was no inflammation? We thought probably the activation there is different. So we tested innate immune cells isolated from the gut and compared with the bone marrow macrophages. There we found that the cells from the gut can be still activated by a LPS or heating activated bacteria. So there is hyper production of beta upon stimulation. So, but yeah, the R18 level didn't show a clear elevation in this experiment. So yeah, if there is no constitutive inflammation, we wanted to see whether to give a DSS challenge to yeah to induce colitis. Maybe there is a phenotype, but to our surprise, we found that the mouse carrying the NAPS gain of function mutation actually have less disease. They have less weight loss, and also we checked many other parameters, including the colon loss and some inflammatory cytokines, and also the permeability of the gut. The NAPS3 mutant carrying mouse have less inflammatory damage in the gut. So we also tested with a tumor model, that's AOM plus DSS induced colon cancer model. In this case, it's again the animal carrying NAPS3 gain of function mutation have less tumor load. We got stuck in this step for quite some time until we found our great collaborator from the Shanghai Jiaotong University. That's Professor Li Ping Zhao and Chen Hong Zhang's lab. We realized maybe there is something to do with microbiota. So we did a co-housing experiment and found that after co-housing, the phenotype in this case is colitis comes into between. So the co-housed animals have some sort of intermediate phenotype compared with lead control wild type or the mutant animals. So we thought most likely there is a microbiota difference, which was confirmed by the microbial 16S based sequencing. We found that the lead control animal and the mutant animal, their microbiota pattern is very different. If we analyze the structure or network between the microbes, the mutant mouse shows such a scattered pattern, but the lead control wild type showed a very dense distribution of the microbes. And after co-housing, they didn't become 100% identical, but somehow they came into between compared with the single mutant versus wild type animals. And accordingly, the disease phenotype also get into intermediate after co-housing. So to nail down the contribution of microbiota from these two animals for the disease, we take feces from either mutant or wild type animals and give to the germ-free animals and then induce colitis. We found that if the germ-free animals were reconstituted by feces from the mutant, there is less colitis compared with the control. So until this step, we found the answer to the early phenomena we found, which means the changed microbiota contributed to the phenotype. Now the question is, how come the microbiota was changed? We thought probably it's through the downstream cytokines of the NAP3 inflammasome. We checked the expression pattern of our beta IL-18 and also NAP3 in the gut immune cells. We found that in the gut immune cells, there is NAP3 and our beta expression, but the pro-IL-18 level is very low. In contrast, in the gut epithelial cells, there is no clear signal for NAP3 or beta, but there is a clear expression of IL-18. So we thought most likely the NAP3 mutant initiate signaling through beta for the downstream effect. Using the animals either untreated or treated with DSS, we also confirmed that our beta generation from the gut is dependent on both NAP3 and caspase-1. Instead, the IL-18 generation depends on the caspase-1, but it's independent from NAP3. Probably it's depending on some inflammasomes expressed in the epithelial cells. We will come back to IL-18 later on. Now we still follow the question we asked, how NAP3 mutant shaped the microbiota. We found that, yeah, there is a big change between NAP3 mutant animal compared with the control in terms of a lot of antimicrobial peptides. And if we cross this mutant mouse with IL-1 receptor knockout animals, so the protective effect is gone, and also the antimicrobial peptide pattern is changed. That means the NAP3 mutant through IL-1 beta induced antimicrobial peptide, and this should have impacted the microbiota composition. So if there is a remodeled microbiota, it can be either protective or destructive. How come in our case is protective? We thought there should be still elements controlled by microbiota that was contributing to the phenotype. We found the regulatory T cells in the mutant animals is more enriched compared to the control or cohort groups. And if we deplete Tregs, the colitis phenotype is returned. And also, if we cross these mutant animals with a rat knockout, which is Treg deficient, we saw there is automatic gut inflammation. Yeah, and a lot of different parameters also showed inflammatory damage in these animals. So let me just summarize with this slide. So in the normal gut, there is a homeostasis status, and if the NAP3 is mutated in the gut immune cells to generate a higher amount of IL-1 beta, it will induce the antimicrobial peptide generation, and then this will impact the gut microbiota composition, which will lead to Treg differentiation. And in this case, if there is a challenge in the gut, so the mutant animals have more of the Treg, so inflammation cannot be easily overwhelmed. But in the case of a normal animal, there will be overwhelming inflammation and leads to colitis damage. Yeah, this is the first story I want to share. And after that, we realized IL-18 is a very special molecule. It was kind of out of our expectation that in the gut innate immune cells, the 18 level is so low, but it's very highly expressed in gut epithelial cells. We checked the database and found in many organs there is IL-18 expression, but in some tumor cells, R18 expression is lost. And it, it's found that in some tumor types, high expression of R18 uh, is positively correlated with a good prognosis. And in colon cancer patients, the R18 expression is very low compared with healthy donor. So we hypothesize that probably losing R18 can be um, a mechanism for the epithelial cells to grow uncontrolled and become tumor. Um, so a few words about R18. So as I mentioned in the very early part, pro-R18 can be cleaved by caspase-1, and recently it's found it can be also cleaved by caspase-4 in a human molecule and generate mature R18. Mature R18 signal uh, through its receptor and induce NK and T cell activation. It is very important to induce interferon gamma and inflammatory cytokines to control tumor. But there is a molecule called R18 binding protein. It can bind the R18 receptor with very high affinity. Uh, which made the R18 in many cases cannot really fulfill its function uh, in vivo. And also, besides caspase-1, caspase-4, it was found some other proteases like caspase-3 can cleave pro-R18 and generate a short form. And uh, quite some years ago, there was a study showed that the caspase-3 cleaved short form of R18 cannot induce interferon production like the mature R18. So this thought this short form is not functional. Um, then we thought, well, maybe this short form is doing something in the tumor context because caspase-3 activation is often found in the um, yeah apoptosis process of tumors. We confirmed that compared with macrophage in the cancer cells, um, there is no caspase-1 activation, and the caspase-3 can be easily activated upon some stimulation. And in this case, we can see there is a short form of R18. Um, so we characterized this short R18 through in vitro experiments and found, if we use recombinant caspase-1, it can cleave and generate a mature R18. But if we use caspase-3, there is a short form of R18 generated. It is a short form of R18. It doesn't secrete into the supernatant like the mature R18, but rather stays in the cell. And if it stays in the cell, it goes into the nucleus. From this staining, we can see the short R18 is mainly in the nucleus. If we do a fractioning of the tumor cell, we saw the short R18 is exclusively in the nucleus fractioning section. And also compared with mature R18, which is binding to the R18 receptor, short R18 doesn't bind the R18 receptor. So the short R18 is not secreted, it's localized in the nucleus, and it doesn't bind R18 receptor. So from this aspect, it's very, very different from the mature R18. But does it do anything in vivo? It goes too much. Yeah, we did a syngeneic tumor model um, by either deleting the uh, R18 in the tumor cells like MC38, which has endogenous R18. If R18 is deleted from these cells, um, the tumor growing is much stronger. And in some other cells like B16, there's no endogenous IL-18. We overexpress the wild type form. It can inhibit the tumor growth. If we introduce some mutant form, which can be not cleaved by caspase-3 or caspase-1, the D69 is the caspase-3 resistant form. In this case, the tumor grow very quick. The D635A is the caspase-1 resistant form. Uh, this one, behaves like the wild type. Um, if we use adenovirus system to specifically overexpress the short IL-18 in the B16 tumor cells, the tumor growth can be strongly inhibited, which is also true in the IL-18 receptor knockout animal, which is telling that this effect has nothing to do with mature IL-18 signaling. So we went further to analyze the mechanism and found that, uh, yeah, in many different types of the immune cells, we found if there is IL-18 expression in the tumor, there is a higher amount of NK cells recruit to the tumor macro environment. And also the interferon gamma producing NK cells is also high in this group of samples. If there is a caspase-3 resistant mutation, this effect is gone. Um, this is telling that most likely, if the tumor cells harbor short IL-18, it can recruit NK cells to inhibit the tumor growth. We confirmed this by the NK depletion experiment by injecting the anti-NK1.1 antibody and found that if the NK cell is depleted, the short IL-18 didn't have chance to fulfill the protective effect. We used also another model that's the AOM-DSS induced tumor model in the IL-18 mutant animals harboring a D69A mutation, which means these animals cannot generate short IL-18. So in these animals, there are more tumors and less NK cells, as we found in other models. So how did this happen? We did RNA-seq analysis using the tumor samples and found that if there is short IL-18 in the tumor microenvironment, there is stronger interferon signaling pathway and also JAK-STAT signaling related molecules. And indeed, if we take the tumor samples and check the STAT1 phosphorylation, the short IL-18 generating, I mean, in the tumor cells expressing Y-type IL-18, there is stronger STAT1 signal. But if there is a mutant where there's no chance to generate short IL-18, this signal is much more compromised. If we check the downstream molecules, RSG genes like CCF2, RSG15, and RF7, they were all upregulated by the presence of short IL-18 in the tumor cells. And in vivo, if we knock down STAT1 in the tumor cells we inject, there is a much quicker growth of these tumors compared with the tumor that harboring intact IL-18 molecule. So we asked the question, how come the STAT1 signal in the tumor cells can induce NK cell recruitment? There should be some molecules mediating this effect. From literature study, we found that the RC15 can stimulate the FFE1 receptor on NK cells to induce NK activation and recruitment. We performed a secretome analysis of the tumor cells, either expressing of short-18 or the control, and found RC15 is enriched in these samples. And in vitro, if we stimulate the tumor cells with short-18 intact, there is higher IC15 generation. And if there is a short-18 in the cells, the STAT1 transcription factor binding to the ISG15 promoter is strongly enriched. That means the STAT1 enhanced ISG15 generation, which recruited NK cells. This point is confirmed by our in vivo experiment. If we knock down ISG15 in the tumor cells, the tumor growth will be much quicker and there is less NK cells. Yeah, as for how the short-18 affect the STAT1 activity, we found that there is no interaction between short-18 and STAT1. But also, yeah, instead, there is a CDK family member called CDK8 is influencing the STAT1 activation status, which is confirmed in our inhibitor study. If we use inhibitor for CDK8, the short-18 enhancing of STAT1 is abolished. And we did further biochemical studies and found short-18 can directly interact with CDK8 through specific regions on the protein. And the final mechanism is that the short-18 can enhance the kinase activity of CDK8 so that the CDK8 can enhance the STAT1 function. So in human patient samples, we also found that the nuclear localization of short-18 is high in the normal tissue, and also if there is a higher expression of short-18, there is better prognosis of these patients. This is also correlated with the STAT1 phosphorylation signal, and there is a positive correlation between the nuclear short-18 score and the CD56, that's the NK cell staining. So if we separate the patient samples into three groups, those who have higher short-18 in the nucleus and also high STAT1 expression have the best prognosis. And if both of these two signals are low, the patient has the worst outcome. Yeah, the others are in between. So if I summarize this part, in the classical understanding of material 18 pathway, macrophage can have inflammatory activity to generate material 18, which can stimulate NK and CD8 T cells to control tumor, but this can be compromised by the IL-18 binding protein decoy receptor. In our findings, we found that in the tumor cells, when there is a Caspase-3 activation, a short form of IL-18 can be generated. It goes into the nucleus, binds CDK8, enhances its kinase activity, and drives the STAT1 phosphorylation at serine 727 site. This activity leads to ISG15 expression, which can induce NK cell activation so as to inhibit the tumor growth. So Yeah, this is our recent finding. Um, and so far, I talked about the gut homeostasis and then the tumor. But back to the microbiota, and also I switched the gear to the lung part. Um, as I mentioned earlier, there is a different microbiota pattern if NAP3 is mutated. Eventually, if NAP3 is knockout, the microbiota pattern is also very different from the wild type control. And yeah, quite some years ago, it has been found that NAP3 is important for host control of influenza or viral infection, as the NAP3 knockout have much severe disease upon influenza viral challenge. And also, it was found that the gut microbiota is important to control influenza. Uh, so we thought probably the gut microbiota can uh, signals through the NAP3 inflammasome to control influenza. Like some other studies showed, microbiota can impact the top interferon for this effect. So we did a simple co-hosting experiment and found that if a wild type animal is co-host with NAP3 knockout, it will get a much better survival upon influenza viral infection. The NAP3 knockout itself, no matter it's a single host or co-host, they have severe disease. So which means in the NAP3 knockout microbiota, there is some good microbes, which is protective against the influenza, but it cannot protect itself. So these are the interesting questions we want to follow. What is a good microbe and why it doesn't protect itself? So for the first question, we did extensive analysis and found probably there are two ASVs or bacterial candidates in the NAP3 knockout was contributing to that phenotype. And we tried and finally isolated one of them. That's ASV4 is finally identified as a bifidobacterium. We gave it a name NJM1. So this bacterium, if we gavage it into the wild type animal, and we found it's protective against the influenza, but it cannot protect NAP3 knockout. And this bacterium, we checked and found that they generate the short-chain fatty acid, the acetic acid. Um, and when we compare the NAP3 knockout and the wild type, the knockout have stronger signal of the acetate. So it's likely that the NAP3 mutant NAP3 knockout animal have these microbes that generating acetate and fulfilling the protective effect against influenza viral infection. And indeed, if we give these animals, the sodium acetate, uh, they are protective against um, IV infection again, and it didn't have the effect on NAP3 knockout animals. For further mechanistic study, we analyzed the lung from infected animal and checked a lot of parameters, and finally found in the mutant animals, sodium acetate treatment during the influenza viral infection leads to strong production of top interferon, but the NAP3 knockout don't have this top interferon generation. If we do this experiment in the interferon receptor knockout animal, we don't see any effect. The animals just die very quickly and don't show any protective effect. of this sodium acetate. Um, so in the isolated macrophages, we found that if we use uh, virus infection and give sodium acetate, the sodium acetate can inhibit the viral replication, which is connected with higher expression of tau interferon. If in vivo we deplete the uh, macrophage from the lung, um, we don't see any protective effect of the sodium acetate, which means the it's likely working on the macrophage in the lung to fulfill this protective effect. For top-line foreign generation, there is a very important element that's the mitochondrial antiviral signaling protein. Upon host cell recognition of viral RNA, mouse get aggregated and then generate tau interferon. So in our experiment settings, we found that the sodium acetate receptor, which is called GPR43, is very important for the aggregation of mouse in macrophages. If there is a GPR43 knockout, we don't see this aggregation, and there will be less interferon generation. Yeah, back to the early questions we asked, why NAP3 knockout was not protected. We found that the NAP3 knockout in macrophage also led to the similar phenotype. If there is no NAP3, then the mouse aggregation is much less, and there is less tau interferon. And if we put all these elements together, we found that the NAP3 knockout or GPR43 knockout, if we use GPR43 ligand or sodium acetate, we found that the mouse protein cannot aggregate on the mitochondria. That explains why there is no tau interferon generation from these cells in the absence of this molecule. So to briefly summarize this part, NAP3 knockout animals generated harbor such a special bacterium, Bifido pseudolongo. It can generate acetate, and through the GPR43 receptor, it recruits NAP3 and mouse to generate a strong signal for tau interferon generation. This is very important to control virus. In the case of wild type, there is no such bacterium, so the interferon signal is kind of weak. But in the NAP3 knockout, there is such a bacteria, but this signaling cascade is broken, so there is no effective generation of tau interferon either. So that means special microbes from the NAP3 knockout microbiota can enhance the host antiviral response through tau interferon. And this is also a study showing NAP3 is not only mediating inflammasome activation, but can also contribute to tau interferon generation, at least under this special context. Well, I guess I prepared too much content, but let me quickly go through the final part. That's the COVID study. So the during the COVID time, we also started some work on this direction, which was from some observation that in the COVID patients that passed away in their lung, there are fused cells. It's called synthetia or cell-cell fusion. And cell-cell fusion is um, a phenomenon found during the uh, yeah SARS-CoV-2 infection. Either the viral particle and the target cells needs the fusion for the infection process, or the infected host cell, can fuse with the neighboring uh, healthy cell to spread the virus. In this case, a neutralizing antibody cannot even target it. So it's a very important element for the viral infection. Okay, if you see this little movie, so this one is viral infected cell. This one is viral infected cell. This is a neighboring cell. If they fuse with each other, then the synthesia is formed and the virus can spread through this mechanism. Yeah, we established a set of systems to study cell-cell fusion. One is to observe the formation of synthesia. The other is to using the luciferase system to show if there is a spike protein and ACE2 mediated fusion, there is stronger signal of luciferase. And also, the cell-cell fusion is associated with cleavage of the spike protein at special site. We call it S2 prime fragment generation on the R815 site, as we found several years ago. And um, yeah, we thought probably the host immune cells can somehow respond to the cell-cell fusion and interfere with this process. Uh, so here we found that if we stimulate the uh, human monocytic monocyte with TLR ligand to get it activated, then the secreted some constants from this cell culture supernatant can inhibit the cell-cell fusion. Here, as you see, there is less luciferase signal, less of the S2 prime cleavage, and less of the fused cells from this observation. And what might be the content of fulfilling this function? We screened some cytokines and found that interleukin-1 beta and alpha were doing the job. So yeah, through the signaling pathway dissection, we found that the R1 beta inhibit cell-cell fusion through the MRD88, ROC, and the TRAF6 part, but it has nothing to do with TAC1 IKK part. So instead, we found the alpha beta can activate the RhoA signal. If the RhoA is activated, which is dependent on MRD88 and TRAF6, there is an inhibition of the cell-cell. There's a compromise of the inhibitory effect of alpha beta. And the reason is that if alpha beta stimulate the the cells, there will be a RhoA activation, and there is an enrichment of this signal between the neighboring cells. I mean, in the control group, the cells tend to fuse together, and the RhoA is distributed around outside of the fused cell. But if we use arm-based treatment, the RhoA signal stuck between cells, and then these cells cannot fuse with each other. And if we do a staining for the cytoskeleton element F-actin, we found the activated RhoA is colocalized with F-actin. And if there is F-actin between cells, they cannot fuse with each other. So using different cells, we saw the similar phenotype. And the ROE signaling, um, let me see here. Yeah, it is still showing the um, same point, but yeah, with a time course. If we treat the cells with interleukin-1 beta, the F actin can stuck between different cells, then they cannot fuse with each other. But in the control group, they just yeah push the F actin outside, and then the cells fuse together. And this signal, I mean, this phenomenon is uh, connected with the ROCK signal. If we use ROCK inhibitor to block this signal, then that effect is gone. Uh, yeah, you see, our beta treatment can make the cells separate from each other. If we give a ROCK inhibitor, they again form fuse the cells. In vivo, if we use a low amount of alpha beta to treat the animal and give SARS-CoV-2 infection, there will be much less of the virus spreading, and this can be yeah turned back if we use alpha beta antagonist treatment. The same is true if we use ROCK inhibitor treatment. Um, that can uh, turn the inhibitory effect of alpha beta around. So to summarize a little bit, the TLR ligand stimulation of innate immune cells can stimulate the IL-1 receptor mediated signal through MRD88 and TRAP6 for ROA activation and ROC mediated actin bundle formation between different cells. Um, so formation of this actin bundle will block the formation of cell-cell fusion, thus to prevent um, virus spreading in vivo. So the a low amount of pro-inflammatory cytokines can be very important to control virus from this aspect if the virus have the mechanism of cell-cell fusion. Um, so I mentioned the integrative immunology as a title. That's my own understanding of our immune system in combating respiratory viral infection and also inflammation or cancer in mucosal tissues. I started all these studies from inflammation and found that besides innate immune signaling in these tissues, commensal microbiota is also connected with these processes. I think probably we need to have an integrated thinking about all these diseases and yeah, um, try to dissect the deep mechanism, keeping in mind the other elements might be affecting the complicated pathological processes. So I yeah acknowledge my coworkers who did all this work and the collaborators from Tsinghua Hospital, Guangzhou Medical University, Shanghai CDC, Shanghai Jiao Tong University, and also yeah Warren from NIH, and also support from different funding agencies. Yeah. I will be happy to take any question. Thank you.